morning, dear students. Today, let us learn about the male reproductive organ of flower in detail. That is, we are going to study about the stamen, its structure, composition, and what it is going to produce. Okay. See, dear students, the process of reproduction in plants can be studied under three headings: pre-fertilization, fertilization, and post-fertilization and embryo development. Okay, so what we are going to study today, the detailed structure of male reproductive organ, the stamen. It is coming under pre-fertilization. Okay, so under the heading of pre-fertilization, we are going to study the male reproductive organ and female reproductive organ, and then process of formation of male gamete in the male reproductive organ, and at the same time process of formation of female gamete in the female reproductive organ. So our topic of today is the stamen. As we know that stamen is the male reproductive organ of flower. Now this stamen comprises of two parts, namely a long stalk which is called as filament and a cap-like structure at its terminal end called anther. Dear students, this filament is going to get attached to the thalamus of flower, or in some cases this proximal end is going to get attached to the petals of the flower if it is a epipetalous condition. Okay, so now to the terminal end a lobed structure called anther is present. In some of the cases the anther is comprising of a single lobe called as monolobate anther. Similarly, we have in majority of the plants dilobate anther or bilobate anther, where it is having two lobes. Okay. Now, let us study in detail by taking the transverse section of anther. See, in this diagram, we are going to observe a three-dimensional view of transverse section of the anther, where you can see the anther wall is slitting or dehiscing, releasing the pollen grains, and you can see the pollen sacs which are going to be dehisced and releasing the pollen grains. Dear students, now this, each anther we can see this is one lobe and this is another lobe. And in each lobe, here also you can observe, this makes one lobe of anther and this is another lobe of anther. Now, in each lobe, we are going to see the two chambers. This is one chamber and this is another chamber. And these chambers are called as theca, in plural thecae. So, in each lobe of anther, there are two thecae present. So, totally in a bilobate anther, we are going to observe 4 thecae. 1, 2, 3 and 4. Okay. So, overall, there are 4 theca present in a bilobate anther. Now, when you observe thus the transverse section of young and mature anther, young anther, we are going to see the anther is covered by 4 different wall layers. Okay, it is covered by 4 different wall layers, and the anther is going to get connected to the filament by a point of attachment, and this is called as connective. Through this connective tissue, it is going to attach to the filament. the stalk of stamen. Okay. Now coming to the detailed study of covering layers of anther and the tissue which is present within each of the theca of anther. See dear students, it is covered by a outermost layer called as epidermis. The epidermis is made up of thin-walled single layer of cells which are compactly arranged that makes the covering of anther. And within the epidermis, we are going to see the endothecium. Endothecium is present next to epidermis inside the anther. The endothecium comprises of fibrous material in the tangential wall and in the inner wall. So this fibrous wall helps the endothecium during dehiscence of anther specially. Okay. Other than that, both epidermis and endothecium along with the next layer called middle layer, they help in covering and protection of the inner sporogenous tissue and microsporangium. At the same time, these three layers, epidermis, endothecium and middle layer, are going to help in anther dehiscence and release of pollen grain. Okay. Now, coming to the next layer of inside the endothecium. See this, the inner to endothecium, there is a layer called middle layer, and the middle layer is made up of sometimes one, sometimes more than one layers of cells. So 1 to 3 layers of cells will make the inner wall of the anther called as middle layers. Okay. Now these middle layer cells are made up of thin wall. Okay. And they have plenty of cytoplasm and nucleus. Usually, along with epidermis and endothecium, middle layer also help in giving protection to the microsporangium present within the anther, and secondly, it also helps in anther dehiscence. And by the time the microsporangium matures and becomes pollen sac, and the pollen grains are formed within the pollen sac, at the time the middle layer cells start degenerating. Okay. Now, next to the middle layer, the innermost covering layer of the theca of anther is the tapetum. Tapetum, its speciality is it is made up of single layer of cells, and the cells comprise of plenty of dense cytoplasm with one or two nuclei. Usually more than one nuclei are present. Prominent nuclei are present within the tapetal cells. The significance of tapetum is it is the nourishing layer. It provides nutrients to the developing pollen mother cell or microspore mother cell, and then its division, meiotic division, for the production of the pollen grains or microspores. So, the tapetal layer cells are going to continuously supply nourishment to the sporogenous tissue present within it. Okay. Now, this inside this what we see is a microsporangium. Okay. So, the microsporangium comprise of a tapetum as the innermost layer, and inside that we see there is a microsporogenous tissue, commonly called as sporogenous tissue. See dear students, during young condition, these microspore cells will be, microsporogenous tissue cells will be polyhedral in nature, and they comprise of a big prominent nucleus with plenty of cytoplasm and nutrients. Okay. Now among these microsporogenous tissue, each and every cell is going to develop into a microspore mother cell, also called pollen mother cell. Okay. So each cell of the tissue is going to become microspore mother cell or pollen mother cell. Now this microspore mother cells, these microspore mother cells, they develop, they grow, develop and undergo meiosis, the meiotic cell division, which will give rise to 4 microspores. Each microspore mother cell divides meiotically to produce 4 microspores, which later grow and develop into pollen grains. Pollen grains. So in the beginning, the microspores will be in the icosahedral or tetrahedral or polygonal shape. So each mother cell will give, pollen mother cell will produce 4 microspores which are joined with each other, making a cluster of 4 microspores in each cluster or bunch called as microspore tetrad. Okay, which later develops or becomes spherical in shape, from polygonal to spherical shape, and at maturity they get separated from each other and they are called pollen grains. And by that time, all these 3 layers, the epidermis, endothelium and middle layer, they are going to burst open and slit. Okay, they slit, that is called as dehiscence of anther. So when the anther dehisces, all the pollen grains from the pollen sac will come out. So at that time, this microsporangium will be called as pollen sac. So in each lobe 2 microsporangia were there, no, in the earlier, in the beginning. Now they unite to form a pollen sac, and from there all the pollen glands are going to be released when the anther bursts open or it dehisces. Dear students, this dehiscence of anther take place in different ways in different species of plants. Sometimes it will open like a lid of the bottle. Sometimes it will slit longitudinally. Sometimes it will rupture irregularly. So in different species, they are going to dehisce in different pattern. So that's about the detailed study of transfer section of young anther. Okay, so I hope you have understood the topic nicely. If you find any doubt, you can ask the question in comment. And if you like the video, then please subscribe it if you are watching this video for the first time, and share it to your friends. Thank you for watching. Thank you.